I guess I'm going to talk to you probably for about 30 to 40 minutes just to give you an overview of some of the stuff that you'll probably see during this week, and to give some context around what is computational physiology and how that relates to some of the projects that hopefully you'll be involved with. So thinking about computational physiology, what are we talking about here? So I'm going to use examples from some of the stuff that I've been involved with, but I'm hoping that you're going to be able to take these examples and apply them to the areas and the domains that you're involved with. But typically we'll have some sort of measurement. Perhaps in this case it's a medical image, so this is a medical image of the heart. It's a little hard to see. But there's a long axis MRI image of the heart. This is the left ventricle, which we have segmented out the outside surface in blue, the inside surface of the left ventricle in green. The right ventricle is around here somewhere, and the atria sit up here. So the idea here is that we have some information. This could be time varying information as the heart is beating, for example, but we're trying to extract out, in this case, some geometric information of the anatomy from our image. We then might have also some other types of measurements as well. So here's an extended volume confocal image down here. This is a section through the rat heart from the outside surface of the rat heart here to the inside surface here. That's about 3 millimeters in dimension by about 1 millimeter square, and what we're looking with these extended volume confocal microscopy images is the detailed structure. So we've stained this tissue using a stain which highlights the collagen structure in the bright colors. The red colors, the pseudo color here in red is to represent the muscular structure, and the idea is that you can see this detailed distribution of collagen throughout the muscle. So again, in this case we have some detailed biophysical information around the structure of the tissue, and we want to be able to measure that and build that into a computational understanding of the tissue. So the other sorts of things we can look at are mechanical properties. So we might have some, in this case, some force measurements, some displacement measurements. We might also be looking at things like electrical conduction. So we might have some voltage measurements and all sorts of different types of functional measurements, I guess, that we can gain from the piece of the organ of interest or the tissue of interest to be able to understand what's going on. And so with computational physiology, the idea is to take all of that information, so rich information on anatomy, on structure, on function, and to build that all together in a consistent mathematical framework, which, because we're engineers, we want that consistent mathematical framework to mean something physically. So we can use the laws of physics to be able to provide an integrated framework whereby we can interpret all of these data in a common physical computational environment. So we want our models to adhere to the laws of physics. I guess that's the idea, and we want to be able to interpret all of this information appropriately using these computational models. So we have physical laws. We also have some empirical relationships that we need to develop, and those empirical relationships need to be fitted to the experimental data that we measure. So we'll come back to some of these ideas later. Computational physiology requires computers, so any idea what sort of code this is? Anyone recognize this sort of code? What language is it? Python? No. Fortran, yeah, this is Fortran 66, I think. So this was, there's the piece of code that was written around here started by the director of the Institute, Peter Hunter. And I don't know if he wrote these lines of code, but this was some of the original lines of code that sat inside a legacy program called CMIS that Peter started as part of his own PhD. So this is old style stuff. You're going to be using much more modern stuff than this. This is a picture of a Coke machine. Actually, no, it's not. It's a high-performance computer. But this was at one stage a supercomputer that similar to the one we were using a couple of decades ago. I think it had 12 processors and some massive amount of maybe 2 gigabytes of memory or something like that. So the idea is that we have software that we write, and there are much more modern languages than this, obviously. We need to solve these things on computers, maybe even on tablets or laptops or high-performance computing, to be able to bring together all of this information using this physical context. And so then we can bring all this together and have models that effectively can predict something about the function of the tissue, whether it be the mechanical or the electrical or the chemical or the energetic sort of function of the tissues. that we're dealing with, the organs that we're dealing with. And the idea is that we would like to be able to gain some biophysical insight, some mechanistic insight as to what's going on with the healthy tissue, and in particular what happens when things go wrong. So what happens when we have a heart attack? What happens when we have breast cancer? What happens to these tissues? How does the function of these tissues change? What happens when we have an electrical arrhythmia? What happens when with the neuromuscular junction in our muscles? How do these mechanistic functions work? How can we describe them? And whenever we're coming up with some sort of predicted function, we would like to also be able to validate that predicted function. So in this case, I've used lots of examples of the heart here, but here's a short axis MRI image of the left ventricle there and the right ventricle here. Here's the long axis image of the same sort of thing. So we get very, very detailed, high quality, high fidelity information that we can use to test the predictions. So this is the sort of idea of, in a nutshell, what computational physiology is about. So the sorts of governing equations that we use, and I'm going to be testing you on this stuff later, so I expect you to commit all of this to memory. We've got different types of equations that we use for different types of physical systems. So we've got reaction-diffusion equations for electrophysiology sorts of problems, flow problems, so mechanical flow, soft tissue mechanics here. So the finite elasticity equations of equilibrium here, this is effectively Newton's laws of motion summarized here. There's some empirical equations, which are the stress-strain relationships, the constitutive equations. Electromechanical equations, sort of Boltzmann sorts of equations. All of these sorts of things come from the laws of physics. They represent the laws of physics. Our challenge is to turn those equations into something computational, so a numerical implementation of these equations, and being careful about how we deal with those numerics, but then building those numerics such that we can interpret that broad variety, that rich variety of information that we talked about on the previous slide. So we can interpret that information very efficiently and effectively. And so the sorts of numerical methods that we often use finite element methods for these sorts of things. There are a whole range of different numerical methods, and so things like finite differences, finite volumes, mesh-free methods, extended finite elements, all sorts of different numerical methods that we can use to take on board the spatial and the temporal information that we're involved with. We might use Hamiltonian mechanics and numerical methods around that to be able to solve one-dimensional type systems. Poisson flow for flow sort of system. So we can simplify these equations and the numerical methods that go along with them to 1D analyses, for example, in the circulatory system. We can do that. So we tend to tailor the numerical methods that we use to the sorts of physical problems that we're solving. In the soft tissue mechanics sense, and the stuff with the heart, we often use finite element methods, 3D finite element methods with anisotropic material properties, because we've got different stiffnesses in the different directions. And so we use finite element methods to do that based upon a Lagrangian approach where we can deal with microstructural material coordinates and in 3 dimensions, in different types of coordinate systems. So we can use those coordinate systems to represent the different fields and the different quantities of interests numerically, so that we can solve these sorts of things on computers. And so I won't go into any detail here, but we tend to build these models based upon these microstructural coordinates. And why do we do that? Well, we're interested in really, if we're trying to describe the biophysical function of, say, a muscle, we're interested in the direction of contractility along that muscle. So we know that skeletal muscles are fairly end-to-end. They have fibers that run end-to-end from insertion point to insertion point. We know that as they are stimulated, those individual muscle cells along those fibers generate contractile tension along their longitudinal axis. So as I stimulate my biceps, I have a one-dimensional force generator along this direction, and I have passive properties. As I shorten my muscle, the volume stays about constant, so the muscle expands in the transverse direction. So there are passive structures which are withstanding that stretch in the transverse direction. I'm interested in how all of those stresses, the active forces, the passive forces in the transverse direction, how that relates to that underlying muscle structure. So the pennate structure of the muscles is important. In the heart, it's really complicated. We've got these one-dimensional structures that rotate from about minus 60 degrees with respect to the short axis image on the outside of the heart, all the way through to 0, all the way through to about plus 80 degrees on the inside of the heart. So those one-dimensional fibers that generate that tension, they have this very steep rotation through about 10 to 15 millimeters. And that's what gives rise, it's that rotation of those muscle fibers that gives rise to the twisting feature of the left ventricle, for example, as it contracts. So we develop our mathematical models to build upon that knowledge of that underlying muscle structure. And we need to use these microstructural material coordinates to do that. Similar sorts of things with electrophysiology. We know that the electrical conduction is fastest along these fibers and it's slowest transverse to those fibers. So we need to build those electrical conductivity, those parameters that we use to represent those physical electrical conductivity features on top of these microstructural material coordinates. We tend not to use spatial coordinates to do this sort of stuff. You know, the coordinate axis of the room, sort of X, Y, and Z up, that's got nothing to do with the function of my arm if I'm facing this way or if I'm facing this way. So the global spatial coordinates are not particularly useful to me when I'm interested in the biophysical function. We need to think about these material coordinates. And so the great thing about bioengineering is that it's really challenging. There are so many different types of tissues that we can do that we can look at different scales. We can look at different types of organs and different spatial scales, different temporal scales, but also different types of physics. So we've talked a little bit about mechanics, we've talked about flow, we've talked about electrophysiology, energetics, all sorts of different types of physics involved, and also at different scales. So all the way from the kind of body scale to the organ scale to the tissue scale, down to the microstructural scale, the cellular scale. So this is a transverse image of the layering structure in the heart. So we're looking end on to those individual muscle fibers. And the interesting thing about the heart is that it's not just this rotation of fibers which is important, but also this layering of the muscle cells into layers of about 3 to 4 cells thick, which slide over each other. So again, these microstructural material coordinates are important down at this level. We can become subcellular and look at the ionic currents, for example, for electrophysiology problems. So the individual ion channels that cross the cell membrane, these are important features that give rise to the spreading wave of electrical propagation along cells and through tissues as well. So this is the really exciting part about the different types of systems that we're involved with studying, you know, mechanics, flow, coronary system, electrophysiology sorts of problems over here, all sorts of physics, all sorts of scales, both temporal and spatial scales. So there's lots and lots of challenge, and this is why it's required really to use high performance computing and computational physiology to be able to bring all of this information together in terms of a common environment. So we would like to be able to have the information down at these subcell scales to be linked all the way to what is happening at the whole organ or even the lifetime sort of scale if we're interested in, say, chronic disease, for example. So to build our mathematical frameworks that link all the way up and down these scales is really important for us to think about how do we share information between these scales, how do we inform the models at the larger spatial scales based upon information that we can measure or the sorts of physical properties that occur down at these smaller spatial scales. So that's a real challenge for us as to how to link these models together and how to build in the experimental data at each of these different scales as well. And so in terms of the MedTech core, computational physiology is really a key aspect of the MedTech core. It's involved with a bunch of the themes and a bunch of the flagship projects that are already underway in terms of the MedTech core. The idea of the MedTech core is that it's really thinking about translational research, both in terms of clinical translation but also in terms of commercial translation, to have impact in terms of the sorts of modelling that we're doing. We would like to see that pushed out in terms of clinical impact, but also how to make more money for New Zealand. So that was part of the goals of the MedTech core. And the idea is that the sorts of examples that we're going to be dealing with are really dealing with pipelines of work from data to clinical observation using these sort of computational physiology workflows. So we're going to talk a little bit about that throughout this week. And so with the MedTech core, this is what the original proposal looked like. So the MedTech core has got effectively 5 themes that you probably know some of the names on this list. There are 6 flagship projects, and they're loosely associated with the themes. I think the first theme has got a couple of flagships, and then maybe the others have got kind of 1 flagship project each. There's also a bunch of technology platforms, so around imaging, around sensing, around actuation, around software and manufacture. So there's a whole bunch of infrastructure that we use in terms of the measurements that we would like to make. But computational physiology really appears in a whole bunch of these places. So there's a dedicated platform around computational physiology, but there's computational physiology basically in most of the themes, most of the flagships, and in other places. There's probably other places here that I haven't pulled out, but where computational physiology really sits at the center of some of the activities of the MedTech core. So it's everywhere. And what we're going to talk about this week, and you're going to go through a bunch of examples. You're going to do some hands-on stuff when Hugh gets the software working as well later in the week. But the idea that I want to just sort of introduce you to this week is this idea of computational physiology in terms of workflow. So that we think about the workflow, I've talked about it a little bit in general terms. We think about this workflow as starting from some kind of measurement, doing some kind of processing of that measurement. So we might have an MR image, for example. We might do some segmentation of that image to enable to build a numerical representation of the important information from that image. So I showed you an example of the long axis of the left ventricle. We pulled out the epicardial and the inner and outer surfaces of the left ventricle using some sort of segmentation. We build a numerical representation, so we have some sort of anatomical representation or tissue structure representation, for example. We then build simulations on top of that numerical representation of the anatomy and the underlying structure. From those simulation experiments, we will then do some sort of reduction operation to pull out the information of interest. So typically these experiments will generate screes and screes of data. We're not interested in looking at all of those numbers. We want to extract out some sort of important information from that which is useful to us in terms of understanding, say, the mechanistic function or the disease processes that are taking place. And then once and also as part of this, we need to also think about how to parameterize our models as well. So once we've gone through that step, we would like to think about creating some sort of prediction. So can we predict, say, take some measurements from someone in terms of a diagnostic procedure and predict what we think is going wrong? Can we think about how to use this as a predictive tool for what is happening down in terms of data that we cannot measure? What is happening down at that subcellular level? Can we predict in terms of what is going on with the mechanisms that take place in places where we're not able to actually make measurements? So can we build that mechanistic insight in terms of making these predictions? And importantly, we need to think about how we test those predictions, and that comes all the way back to our experimental observations as well. So there's a sort of general workflow that we're going to step through with some specific examples over the course of this week, and I'll talk very briefly about those examples. So I think that one of the goals of this week, or some of the goals of this week really, are to just give you some idea of the sorts of things that are involved with computational physiology, the sorts of skills that you require, the types of methods that are involved, and the different capabilities that we have across the MedTech core in terms of computational physiology. So the idea is you're going to step through some workflows to understand the typical steps involved, as I've just talked about on the previous slide. We're going to go into some detail for specific examples with those workflows. And the key thing is it's not about really gaining, detailed knowledge about a specific type of tool. We're going to focus on the sorts of tools that we've developed here as part of the MedTech core and with our history of developing this sort of modelling. But it's not really about teaching you guys how to learn that stuff. That's a different sort of set of tutorials that are involved. This is really just talking about the types of things that you need to look out for. What are the sorts of skills that you need? And if you find that some of this resonates with some of the stuff that you need, then you can go and get specific help on those individual tools which is relevant to your projects. So yeah, just giving you some idea about what sort of tools are available. what sort of capabilities there are, what sort of things do you need to think about when you're starting to get into this field. So that's what we're going to try and achieve over the next little while. So this is the bit that might change around a little bit this week, is that right, Andre? Well, in terms of the days at least. The actual modules themselves are going to be about right, but
they'll be in that order.
They will be in that order. The talks are going to be different. Yeah, okay, of course. So these are the sorts of things you're going to do. In terms of that general bubble workflow that I showed you, you're going to step through some actual segmentation of the heart in this case. You're going to look at model construction. I think you're going to have a go at some breast images with that, some sort of simulation, and the application is going to be on the GI tract, some visualization tools around respiratory mechanics, and then workflow looking at musculoskeletal applications, probably using the MAP client. Is that right, Hugh? Yeah. So then you're going to have on Thursday, you're going to have a kind of a extended project that you're going to look at for that day, which hopefully brings together a whole bunch of ideas around this, 2 different projects in fact, which hopefully sort of demonstrates some of the ideas in a general sense around some of these concepts. And then Friday morning, Peter Hunter is going to give you an introduction to using CellML and an important piece of software called Open Core, and you're going to run through some lecture on the sort of cellular ODE style modelling here, and run through some tutorial on how to go about setting up these ODE sort of models. And then, yeah, you'll see where that takes you in terms of Friday afternoon. So the different workflows that you're going to work through over the next few days are in this order. And I'm going to just talk very, very briefly and give you the high-level overview of these workflows. So the idea with the first workflow that you're going to get some experience with or some exposure to is this thinking about computational physiology of the heart. So I've talked about it in some detail already, the idea of taking some sort of medical imaging, going through a set of processes now, so looking at an image segmentation sort of step. So there's a whole bunch of different tools that we can use for this. But the idea, I showed you this picture already, taking a medical image of the heart and extracting out some sort of anatomical shape, anatomical structure for that heart. We can then do that for different individuals. So here's a left ventricular model which is fitted to experimental data for a normal heart. This is a heart failure case here, so the classic signs of extended or decompensated heart failure are large hearts with thin walls. They lose their mechanical advantage in terms of the ability to pump blood. So the ejection fraction here, the cardiac output reduces, the ejection fraction reduces, and this is sort of an end stage state where the person is going to need some help before long, or things aren't going to turn out too well. So the idea is that we can set up these different sort of models based upon these images and look at the different function. We can also build in the hemodynamic kind of response so this is just a kinematic sort of measurement in terms of this subject-specific modelling. The idea is that we can build in the biomechanics now by thinking about the loading conditions. So what are the left ventricular pressures in these cases? They have a big impact in terms of the function. So with that sort of measurement, we can simulate the actual finite deformation elasticity from, say, a relaxed state of the heart to the fully contracted state of the heart, and matching those pressure boundary conditions, loading that up to understand something about the total mechanical function. In order to do that, we need to calibrate these models, so we can use constitutive equations, and there's a whole bunch of different experimental data and theoretical frameworks that are required for these constitutive equations. So I can look at this and I understand that this is an equation that represents transversely isotropic mechanical properties, but this may not be the case for the different types of tissues that we're looking at. In terms of the heart, we tend to use orthotropic material properties, so we tend to extend upon these sorts of equations, both for the passive properties but also for the active properties. And the important thing about these equations is that they have parameters associated with them. So the C2, C3, TCA, beta, these are parameters that we need to be able to estimate for the particular patient or the particular bit of tissue that we're interested in for the heart here. So once we've done that calibration step, we can then predict something about the function. So on a regional basis with the heart, we can look at these local work loops. We can look at the regions of the tissue which are performing useful work. And the loops will be going generally counterclockwise in that sense. We can look at the regions of the tissue that are doing not useful work, or in fact are being carried along as passengers. So they might be subject to a regional ischemia or something where they're not generating contractile tension, they're just being pulled and stretched in different directions. And that might look like clockwise related loops in these stress-strain, fibre-strain sort of plots on a very, very localized level. So then we can make some sort of prediction about the state of the heart, the mechanical state of the heart, the energetic state of the heart, that we can potentially feed back to the clinical community. So that the idea of this workflow, and then you're going to see workflows that look very similar to this. The idea is that we're going all the way from image or medical kind of information, hemodynamic information. We're using the laws of physics here with some calibration to come up with some sort of prediction and feed that back to the clinical community. That's the idea of this sort of workflow. I think he's right there. Yep. So this is the first workflow that you'll get some sort of access to. This workflow, you're going to be just focusing on the image segmentation part. You're going to segment some images and pull out some left ventricular models, I think. And so the image segmentation will be looking something like looking at the, you'll probably be looking at MRI. You can get medical images in many different formats, so cardiac CT, for example, as well, or ultrasound. The idea is that you're going to be segmenting information from these images and look at the data transformations that are required and able to do that. So in this case, you've got different images along different axes, so you've got long axis and short axis images. We need to register all those images together and pull out the geometric information and the functional information, in this case the beating heart information that's going to be interested. So that's what you're going to do with the first workflow. The second example that you're going to come across this week is looking at this workflow for helping with breast cancer imaging. So again, exactly the same sort of idea. The idea is making some kind of medical image here. So this person is lying prone with her breast hanging down in these specialized coils. She'll go into the MR magnet in terms of 3D MR of the breast, looking for cancerous tissue effectively, or even increased regions of increased blood flow. For the MRI case, it might be X-ray mammography if they're looking for dense tissues like microcalcifications or potential tumours. But the idea is that you've got these measurements. Again, there's an image segmentation step, just like we did for the heart. We're pulling out anatomical information. This person's got a very, very large cancer here. But we're pulling out information from the skin and the pectoral muscle surface. We might have the rib muscle surface. So compartmentalizing the tissues into their different tissue types. So the fat and the fibroglandular tissue as well in this example. Building three-dimensional models of the anatomy, so there's patient-specific modeling sense. Performing some sort of simulation with those models. So here we're taking images from a measured prone position to a predicted supine position, and this might be for something like needle biopsy or second look ultrasound. So typically they take the MRI, they'll maybe see something in the MRI that they would like to then follow up with ultrasound. For ultrasound imaging, the person's lying on their back. MRI, they're lying on their stomach, so the breast is in a very, very different position. We can use the laws of physics to be able to predict what does the breast look like, what shape does it take on, how do all of those internal tissues move with respect to each other as we turn the patient on their tummy to on their back. Similarly, for X-ray mammography applications, the person is standing up but their breast is being compressed, really large degrees of compression, somewhere around sort of 50% compression. So these are really large deformations. Again, we can take the information that we gain from the MRI and predict where do those tissues end up during that compression. And we can compare then and set up a registration process to look at information from MRI with information from X-ray mammography. So X-ray mammography is a single 2D image through the compressed breast. We may see a feature of interest, which may be some sort of dense tissue. That dense tissue doesn't show up automatically in the MRI, but what the MRI does show and can show is regions of increased blood flow, for example. So if we can co-localize the increased blood flow from the MRI with the dense tissue from the X-ray mammography, then we've got greater indication that perhaps we've got tissue of interest here that we really should have a look at. There's potential for tumour. Tumours need blood flows to be able to survive. They are very, very dense tissues, usually in breast cancer, clusters of microcalcifications which you cannot see on MRI, but you can see on X-ray. Those clusters are early signs of breast cancer. So again, the idea here is to have this common environment where you can bring together, using the laws of physics, bring together the information from these different imaging modalities. Once we have that registration framework, we can present that back to the user using a web-enabled sort of technology, so WebGL or some sort of browser-based or bespoke kind of application that presents this information in some useful way back to the clinical community. So there are graduate projects in each of these different aspects, and what you're going to play around with, I think, in terms of the workflow for this week is thinking about how to set up anatomical models based upon what you've done in terms of image segmentation. And so thinking about the model construction technique, you're going to start with some data, and then I think this movie is a bit jerky, but it's going to show how we can take those 3D data, take some sort of template model and fit those template models to the data. So there's all sorts of different concepts and things that you need to look out for when you're doing this sort of thing. You need to have these template models, they need to align with the data that you're dealing with. You need to think about how many geometric degrees of freedom do I need to be able to represent these dense segmented data, these green spots, for example, that I get from my images. I'm basically parameterizing that using a small number of geometric degrees of freedom using these finite element models. So how many is enough? How many elements are enough? How many nodes are enough? Those sorts of things when we're fitting these geometric models. So there's a alignment step, there's a sort of a mesh convergence step and a fitting step, all of these sorts of things that you're going to get some sort of aspect to, introduction to. So then the third workflow is going to be looking at the gastro-intestinal tract, and again it's presented in a slightly different format here, but the concepts are the same. Again, you start with some images, some sort of data, do some sort of image processing to pull out the information of interest. Again, setting up the models here using a range of different tools, setting up these low dimensional sort of reduced parameter representations of those detailed experimental or measurements. Then setting up a simulation based upon that anatomical representation and coming up with some sort of calibration exercise, but importantly some sort of prediction or some sort of insight as to really what's going on with this physical system. And so for this workflow, you're going to be, well, the idea is that, I forgot to mention here, just to say that there are instrumentation associated with this this particular project. So the group associated with this up on level 7 here, they're developing specific instrumentation to help make these sorts of measurements to do this sort of prediction. So it's this combination really of the mathematical models, but also the types of experimental data that we can generate to be able to calibrate those mathematical models. So specific instrumentation is often required to be able to get the sorts of measurements that are required to do this sort of calibration. And so for this workflow, you're going to be focusing on the simulation aspect, and I think Andre's going to take you through some of this stuff. Here you're looking at an electrical simulation of the stomach, and we're looking at the pacemaker site here, which is giving rise to these slow wave activities of depolarization down through the stomach here. I'm not really sure what's going on up around here, but this is something that someone else can perhaps tell us about at some point. But the key point here is that we are representing now this physical property, this spreading wave of electrical activation. We're using the laws of physics to be able to represent and set up these mathematical models. We need to think about how we can set up the numerical methods which are appropriate for this sort of simulation. How do we take into account the different spatial and temporal scales that are involved? How to think about really important concepts to do with how do we set up computer models which are not just sort of sledgehammer throwing together 1,000,000 degrees of freedom. What's the balance? Because that's going to take us, you know, a decade to be able to get a simulation of any youth on the best supercomputer. how can we balance the idea of how accurate is enough, how good is good enough with our simulations, such that we can get solutions to those simulations in a reasonable amount of time. I used to think that sometimes a reasonable amount of time is going, how long does it take to go and get a cup of coffee, but perhaps set it going one night, come back the next day and have a look at your simulation results, even on supercomputers as well. Anything more than several days, you have to really be careful about how you organise your data and how you organise your thought process as well, because you come back a week later and it's like, gee, now why did I set that simulation going again? What was the question that I was asking when I've got 20 different simulations kind of going at the same time? So there is this really important concept with these mathematical models of biology. They get really big really quickly. So I think it is important to be able to think about how efficiently can we solve these sorts of problems, and how accurate is enough. How can we determine, based on the sorts of experimental data or the sorts of clinical predictions that we want to generate from these models, what is good enough? And that's the sort of thing that hopefully you'll get a little bit of insight to for this particular workflow simulation task. Okay, so then the fourth workflow that you're going to get an introduction to is thinking about the respiratory system, so computational physiology of the lung. Again, it's presented in a slightly different way, but the idea, we have measurements of different types of features, so that the structure of the lung, we might have something to do with clinical imaging, so CT imaging tells us something about the structure but also the function of the lung as well. The idea is that we can build mathematical representations of those detailed data, so looking at, say, respiratory mechanics or pulmonary blood flow, for example, and how we can combine those different functional representations of the experiments using a combined mathematical modelling kind of integrated setup. So here's the representation of the anatomy and the function of the measurements that we make using this sort of low-dimensional finite element model approach here, or one-dimensional branching structure approach over this side. Again, we can go through some simulation and validation, calibration sort of step with this using a range of different software tools. So looking at, say, gas mixing, we can look at expired flow and the contents of the expired flow in comparison to the inspired flow. Different types of imaging that we can use to be able to validate and test these sorts of models as well. And then setting up interfaces to help the clinical community to be able to integrate all of this information and come out with some kind of insight as to what is going on with that patient's lung, or potentially what is happening, how do we understand the integrated function of the lung using different teaching tools as well. So again, it's a similar sort of approach where, you know, experiment to model, the anatomical model to simulation model to calibration, and then some kind of interaction with the reduced results that we get from these models. And the idea here is that you're going to start to look, I think, at different ways of visualizing the data. So you're going to get some experience in different visualization tools, how to deal with large quantities of data in different sense, to show the different features of interest, if you like, both in terms of the spatial fields but also the temporal fields in terms of blood flow or blood pressure down in this sense. Maybe looking at things like pathology as well. What do they look like? How do you represent these things? How can you portray this information back to the intended audience? Okay, so there's some other key concepts which hopefully you're going to pick up along the way as well, that will be, they're not specifically part of the main workflows, but they will be referred to, I assume, right, Andre? Hopefully. I mean, these are other key concepts when we think about computational physiology. This is a really important concept. The idea is that whatever you do must be reproducible. So thinking about the sorts of standards that are involved, certainly on Friday you're going to be using this, a set of models from a library, a CellML related repository. And so the idea is that languages and standards are being set up to be able to share information between different physics, for example, between different computational platforms. So being able to encode that information in a reproducible format which is downloadable, rather than just writing a model down on in Microsoft Word or something, sending it off to your publication, and you've probably got some errors associated with the way you've typed that model up. you get it through publication, it goes through reviews, you found some errors and it gets published. And I've done this with a paper that I wrote, or a couple of papers that I've written, or you find out, someone emails you typically to say, what happened with that equation, what happened with that parameter, and it's like, oh no, there's an error. So a better approach is to actually use standards to be able to mark up these, what the equations that you use to solve your simulation. You would like those exact same equations to come from the same source as the thing that's going to go into your paper. So you can have a single source, which is the thing that you generate your simulation model from. You can also generate the mathematics, the PDF type mathematics that goes into your paper. If those two things come from the same source, then you can at least reduce the chance of these silly little errors sort of plugging in. So that's a really important concept about reproducibility. Non-ambiguous, well-described, repository sort of information, and how to deal with management of those repositories as well. So where do these models come from? What data are they built upon? What sort of curation has been done on individual models? Who has checked that they actually do what the paper says that they do? So thinking about versioning of models, thinking about where did they come from, the provenance of models is really, really important. And hopefully you'll get some idea or some introduction to this sort of stuff as we step through the week. Okay, so yeah, it's not explicitly part of this module, but it's really important stuff that people need to think about when they're getting into computational physiology. So these are the sorts of things that you will probably, that some of this stuff will be happening behind the scenes. Oh, I forgot to update that. That's got to have an IO on the end of it, I assume. Is that right? It still gets there. But yeah, so you'll be getting access to some of these repositories. You'll be using the Physiome project repository on Friday, I guess, if not before, for some of these sorts of models. We tend to use GitHub around here, I guess, for storing code, for versioning code, and we've got group projects sort of working on the same thing. This is a good code repository sort of system. And then there's other types of repositories that you can use for data and other stuff. Okay, this is the one I did update. So I think there's going to be a bunch of tutorials, interactive tutorials with those 4 different workflows that you're going to start to deal with. We do appreciate your feedback, and up here there will be a link to an anonymous feedback form, which at the end of the week would be great if you're able to let us know what worked well for you and what didn't work so well. Nina's, I think, also going to grab some maybe other types of feedback from you at some point, but we're really interested in how to improve this course for future people who want to take the course. I think that's about it for now. Is there any questions to do with the sort of stuff that I've talked about? Have I covered everything I need to cover? I think so.
We'll find out at the end of the week.
So will you share the presentations?
Yeah, I think...
Or just a movie?
The PowerPoint slides, I think, are available somewhere.
We hope to, yeah. We still have them from the last time. We will try.
Yeah, I thought we put the PowerPoint slides somewhere on Figshare or something,
yeah,
but
just haven't told anyone
how to find them. Okay. Yeah, there was some issue around how we used Figshare last time, if I remember rightly. I think Andre set something up, and then I was unable to update it or something. I don't know.
We have to be careful some of the images we use in those slides as well. For public domain, we don't have the rights to others.
Oh, that's what it was. That's what, yeah.
We have to check those.
Oh, right.
So it's one of the reasons why we're doing that.
Yeah, great.
That's right. Any other general questions to do with the stuff that was it understandable? Was it at the right sort of level to kick things off for this week? Were you able to resonate with something to do with your own project? Because we try to, I mean, my background is on cardiac mechanics and breast cancer imaging, so you could probably see that I spoke a little bit more detail around that sort of stuff. But, you know, I have worked on musculoskeletal projects and other projects as well. But we do try to make this, we try to target the different areas to make it applicable to your projects. So if you've got any feedback on that, that would be helpful as well. Anything to do with computational physiology, at least, anyway. Okay.